I am a graduate student in Dr. Nana Bursak's lab at Duke, and I'll be talking about, like Dr. Camp said, our work looking at targeted ERK activation within our in vitro system, neonatal rat ventricular cardiomyocytes. So as our previous speaker mentioned, there exists some models of regeneration in the heart, including neonatal mice and zebrafish, which both after injury are able to encourage endogenous proliferation of surviving cardiomyocytes near the injury site, which results in a restoration of heart function and essentially a normal life. But as we all know, probably in adult mammals, and shown here in adult mice, after a heart attack, you instead get a scar formed after injury, and instead of restoration of function, you get a permanent decline in contractility, which essentially the field wants to utilize cardiac mitogens identified in these first two systems and see if we can encourage endogenous regeneration in adult mice using viral application of these cardiac mitogens, or kind of another method people are using is application of exogenous cells. But today I'll be focusing on encouraging endogenous proliferation. And so shown on the left are many of the cardiac mitogens that have been identified. And something that we noticed, and looking at this, you can see that many of these mitogens are either targets of MAP kinase or ERK signaling, or are activated by MAP kinase signaling. And so we think that while the field has established that ERK signaling is necessary, or at least intact ERK signaling is necessary to drive cardiomyocyte proliferation, what we wanted to ask is whether or not ERK activation alone was sufficient to drive cardiomyocyte proliferation. And to look at this, we overexpressed a constitutively active BRAF mutant, shown here, using a lentivirus, which uses a muscle-specific MHCK7 promoter to drive expression of this transgene along with the mCherry reporter. And our controls for these experiments is just expressing mCherry. And so one of the first things we wanted to look at is whether or not this ERK activation drove hypertrophy. As people have shown previously in vivo, ERK activation in mice leads to larger cell size. But when we look at expressing the effects of expressing this constitutively active BRAF in our neonatal rat cardiomyocytes in vitro, we don't see increased cell size shown here with using imaging data, sarcomeric alpha-actinin used to identify the cardiomyocytes. And in addition, we confirmed this with flow cytometry analysis, showing again that we did not see increased hypertrophy. So what you might have noticed from those previous images is that the expression of constitutively active BRAF seems to negatively affect sarcomere structure. And so here I'm showing both 1 week and 2 week old cardiomyocytes, neonatal rat cardiomyocytes, showing the sarcomeric alpha-actinin staining in green. And what you see is nice cross striations at both time points in the control group. But then with constitutively active BRAF, you see what we think is a progressive loss of sarcomere stability. And something we noticed is potentially an increase in stress fiber formation. And so moving forward past our monolayer system, we wanted to look at the effect of this in a more mature model and one where we can measure function. So just to introduce our model system, we have neonatal rat cardiomyocytes, which we embed in a fibrin-based hydrogel and culture generally for 2 weeks. And what we see are contractile, essentially cylinders of ventricular tissue. Shown here is the longitudinal view of this tissue showing cross-striated sarcomeres. And in cross-section, you can see that we have cardiomyocytes in the center of our tissue along with fibroblasts along the outside stained with fomentin. And so for these experiments, looking at expression of BRAF, we applied lentiviral overexpression of the transgene on the day of tissue formation and cultured for the 2-week time point and added EDU for 48 hours prior to collecting the tissue in order to analyze cell cycle entry. And so one of the first things we noticed looking at the morphology of these tissues, comparing control on the left and constitutively active BRAF on the right here, is that the size of the tissue appears to be larger with a acellular core forming, essentially due to the increased size inhibiting diffusion of oxygen. And what we see is also increased momentum, kind of what looks to be in the cardiomyocyte region of the tissue, not just in the fibroblast outer area. And what you can see is when we looked at the whole bundle imaging longitudinally, we see cross striations at 2 weeks as expected, but then we don't see intact sarcomeres, or at least a significant portion of cardiomyocytes show disassembled sarcomeres at this time point, similar to our 2-dimensional studies. And so quantifications of this are shown on the right, showing increased EDU incorporation in cardiomyocytes, increased cross-sectional area or size of the tissue, and increased vimentin abundance. And so, as I mentioned, we can use this system to look at the contractile function of these cells. And so, using a custom force measurement system, what you get here is an example force trace showing our controls in blue and then the constitutively active BRAF in the brown. And what you can see is that the BRAF overexpression results in a decline in tissue function at this 2-week time point. And what we also notice in it is an increase in the passive tension of the tissue, which could be related to the increased stress fiber formation that I mentioned previously. And so we kind of looked at this phenotype in addition to the 2-week standard time point for our group. We looked at it in 1 through 4 weeks to kind of look how this phenotype progresses over time. So what you can see on the top row here is the contractile properties of the tissues, showing significantly lower contractile force generation induced by the BRAF overexpression, as well as increased passive tension which is maintained over time in culture. And kind of the morphology of the tissue, what we see is that there's progressively increased nuclei per cross-section, as well as maintenance in tissue size and increased EDU incorporation over time. And so, as I mentioned, we looked at this model, this BRAF overexpression, to try to see what the effects of activating ERK is in these tissues. And so we, of course, confirmed that ERK was activated at both 1 and 2 week time points in our bundle system. So using Western blot, we see that there's increased ERK abundance and phosphorylated ERK abundance relative to the control groups at both time points. So we are seeing chronic ERK activation as a result of the expression of this transgene. After seeing that, we then wanted to look at whether or not inhibition of MEK or ERK signaling would be sufficient to restore function of the tissues. But instead, what we found is that, so shown here on the left is looking at the sarcomere structure again, significant portion of the sarcomeres are disassembled with the BRAF tissues, but following addition of ERK or MEK inhibitors, we don't see restoration of sarcomere structure, which is kind of related to the inability for these inhibitors to restore function in the tissues. But what was interesting is that we did see decreased EDU incorporation, suggesting that we are inhibiting some ERK activity, which is required for the proliferation induced by constitutively active BRAF, but potentially we are not inhibiting it enough to see a restoration of contractile function. So following these studies, we then wanted to look more mechanistically. So to do that, we performed bulk RNA sequencing, and shown here is our PCA plot with our 1-week BRAF samples here, our 2-week BRAF samples here, and then 1-week controls and 2-week controls in the purple. And so you can see the time points tend to cluster together. But when we look at volcano plots showing the differentially expressed genes, we see over 2,000 differentially expressed genes using this cutoff, comparing the constitutively active BRAF to control at both time points. And so we first looked at the pathways which were implicated in our previous studies. So looking at proliferation, MAP kinase signaling, ECM production or remodeling, and kind of cardiac tissue development, we see kind of the expected trends in expression relative to the control group at both the 1 and 2 week time points. So this kind of confirmed our previous functional studies. We then wanted to look at kind of the most differentially regulated processes. And so using gene ontology analysis, again, we saw that many developmental pathways were upregulated due to the BRAF expression. And one that stood out to us was the regulation of locomotion. And so we wanted to confirm this functionally using live imaging. And so this is a NRVM or neonatal rat cardiomyocyte monolayer culture. We imaged for 6 hours. And this is just showing the nuclei over that time period. And what you can hopefully appreciate from this and from the tracking is that it looks like the proportion of cells which are migratory appears to be increased as a result of the BRAF overexpression. And we confirmed this with quantifications showing the displacement, the distance that the cells traveled from start to end, and the linearity of those migration patterns. And shown here is our isolation purity. So, well, we can't say for certain that the entire difference in migration is caused by differences in cardiomyocytes. We do believe that because our purity is about 90% or higher, we do think that a substantial portion of this difference is in cardiomyocytes. So looking at that, we looked at the downregulated processes, which suggested drastically downregulated metabolic processes, specifically aerobic respiration. And so what we wanted to look at is functionally whether or not glycolysis was upregulated to compensate for this. So to do that, we looked at the concentration of lactate and glucose in our culture media from the 1-week-old cardiac bundles transduced with the control virus or BRAF virus. And what we see is that the abundance of lactate is significantly increased as a result of BRAF expression, and the concentration of glucose is significantly decreased, suggesting that glycolytic metabolism is activated by the BRAF overexpression. What was interesting is that when we went to begin looking at the oxidative metabolism function, we first looked at mitochondrial abundance using mitotracker dye and flow cytometry analysis. And what we found is that in the BRAF cells, kind of the opposite of what we expected to see actually is increased mitochondrial content, which we speculate could be due to the cells compensating for less functional mitochondria per unit by producing more mitochondria. And so we want to look more into this in the upcoming studies. So to summarize, what we found is that ERK activation in vitro drives broad transcriptomic changes, which leads to increased tissue stiffness and reduced contractile function and sarcomere stability, as well as increased cell cycling and migration, and a shift in metabolic function to favor glycolysis. And so with that, I'll thank everyone in the lab and our generous funding sources, and I'm happy to take any questions.